On November 10, 2015, a 46-year-old magazine art director named Laura Huber died in a hospital bed in Silicon Valley. She had stage 4 colon cancer. It had spread to her liver, her chest, her neck. When they found it, it was already too late. 30 days later, her husband Jeff walked into a board meeting at the DNA sequencing company Illumina. He sat down, and he agreed to spend the rest of his career building the test that might have saved her. On January 10th, the company was announced. On February 10th, he became its CEO. The dates were all tens. He has never said if that was on purpose. 10 years later, in October of 2025, that test, called GRAIL, published results showing it could find cancer 7 times more often than every screening test you have ever had. Combined, 50 cancer types, 1 tube of blood. Before your body has the first idea, something is wrong. This is how it works. And why? Statistically, the cancer that might kill you in 20 years is almost certainly in your bloodstream right now, writing its own name in a language we are finally learning to read. Here's something you were never taught. Every cell in your body right now is shedding tiny fragments of DNA into your bloodstream. Not a few, not occasionally, constantly. A cell dies, a skin cell, a liver cell, a lung cell, and its DNA breaks into little pieces. Those pieces circulate through your blood for a few hours before your body recycles them. Scientists have a name for them: cell-free DNA. In a healthy person, most of that DNA comes from normal, everyday cell turnover. But if there is a tumor growing somewhere, even a tumor the size of a grain of rice, buried deep in your pancreas, or your ovary, or your liver, it is shedding DNA too, cancer DNA, into the same bloodstream, circulating past your arm, right now. The signal is there. It has been there the whole time. The only question, for the last 50 years of oncology, has been whether we could ever learn to read it. Most people think cancer gives you warnings: tiredness, a lump, weight loss, a cough that won't clear. For a few cancers, that's true. Breast cancer, you can feel a lump. Colon cancer, eventually, you see blood. Cervical cancer, a pap smear catches it. Lung cancer in heavy smokers, a CT scan catches it. Those 4. Plus a little early-stage prostate screening. That is it. Everything else: the pancreatic tumor growing quietly behind your stomach, the ovarian tumor in a cavity with no pain receptors, the liver cancer that slips in past a healthy-looking liver panel. Those get found because someone is suddenly in pain, or suddenly yellow, or suddenly 30 pounds lighter. And by then, almost always, it is stage 3 or stage 4 pancreatic cancer diagnosed today. 85% late stage. Ovarian, 70% late stage. They are called the silent killers for a reason. But here's the thing: they were never silent at the molecular level. They have been shouting into your bloodstream this whole time. We just didn't have the ear. So what are we reading? Not the DNA itself. Every cell in your body has the same DNA. Your lung cell, your colon cell, your brain cell, genetically identical. If you just looked at the letters, you couldn't tell them apart. What is different is which genes are turned on. Your body does that with little chemical tags called methyl groups. Small molecules, 1 carbon, 3 hydrogens that clip onto the DNA and say, this gene stays off in this tissue. Lung cells have 1 pattern of tags. Liver cells have a different pattern. Pancreas, kidney, bone. Each tissue has its own fingerprint, written in thousands of these methyl marks. Cancer cells have a different pattern again. Distorted, chaotic. When a tumor cell dies and releases its DNA into the bloodstream, that DNA still carries its methylation pattern, its tissue fingerprint, and its aberrant cancer pattern fingerprint. So if you could read every fragment of free-floating DNA in a tube of blood, and you could sort them by their methylation patterns, You could theoretically say, there is cancer pattern DNA in here, and it looks like it came from the pancreas. By 2015, DNA sequencing had become cheap enough to do that, not for one gene, for every fragment, millions of them, in one blood draw. The tools finally existed. The only thing missing was someone willing to bet everything on it. Laura Huber was 46 years old. She was a magazine art director. People who knew her said she always carried a sketchbook. Wherever she went, there were drawings. In 2014, she was tired. That was the only symptom. No pain, no weight loss, no family history of cancer. Fit, energetic. Her doctor found a shadow on a scan. Then more shadows, colon cancer, metastatic to the liver, to the abdomen, to the chest, to the neck. Stage 4 on the day of diagnosis. 18 months of treatment, surgery, chemotherapy, more chemotherapy. The kind of hope that narrows one scan at a time. On November 10th, 2015, she died. Her husband sat with her sketchbook, a thing that had been full of her mind the day before. Now, just a closed book. Here is the part the technology writers usually skip. While his wife was dying, Jeff Huber was on the board of a DNA sequencing company. He was sitting in meetings where researchers were saying, "We think within a few years, we will be able to catch cancer signatures in blood at stage 1, before the patient feels anything." He was hearing the science that could have saved her while it was failing to save her. 30 days after she died, he took the meeting. So what, physically, is Galleri? It is a tube of blood. 20 milliliters, about 4 teaspoons. You hold out your arm. A phlebotomist draws. The tube goes into a courier bag and rides to a lab in North Carolina. There, a machine called a sequencer reads every fragment of cell-free DNA it can find. Millions of fragments per sample. For each fragment, it does not just read the letters, it reads the methylation pattern, the chemical tags, then a software model, a machine learning classifier trained on hundreds of thousands of patient samples, cancer and non-cancer, asks a single question. Does this collection of fragments look like someone with cancer? Or someone without? If the answer is yes, the test goes one step further. Based on the methylation patterns of the cancer signal fragments, it predicts which tissue the cancer probably came from. It is right about 90% of the time. You get 2 words back: cancer signal detected, plus a tissue prediction: pancreas, ovary, liver, head, and neck. And then, and this is important, you go to a real doctor. Who does real imaging? Who finds out if there is really a tumor there? The blood test is not the diagnosis. It is the flashlight pointed at the part of the room where the diagnosis might be waiting. In October 2025, at the biggest cancer conference in Europe, in Berlin, GRAIL presented the results of a trial called PATHFINDER 2, 25,114 adults over 50. Half got standard screening, mammograms, colonoscopies, the works. Half got standard screening plus Galleri. Adding Galleri found cancer 7 times more often than standard screening alone. Let that land 7 times. More than half of the cancers it found were in early stages, stage 1 or 2, which is the stage where treatment still works, and the positive predictive value. The chance that a cancer signal detected result actually means cancer was about 62%. Then there was the other big trial, NHS-Galleri. 140,000 people in the United Kingdom, one of the largest randomized cancer screening trials in history. It found cancer 4 times more often in the blood test group than in the usual care group. It reduced the number of stage 4 diagnoses substantially. But here's the honest part. It also technically missed its primary endpoint. The pre-specified statistical threshold for late-stage cancer reduction did not hit. A critic says, the trial failed. A supporter says, the trial did exactly what it set out to do. Fewer people diagnosed too late, just not by the precise number in the original math. On January 29, 2026, GRAIL filed with the FDA. A decision is expected in 2027. If it is approved, Galleri will become the first FDA-approved multi-cancer early detection test in history. Here is what the press releases don't tell you as loudly. Out of every 100 people tested, about 1 gets a cancer signal detected result. Of that 1, about 62% actually have cancer, which means about 38%, nearly 4 in 10, will be told their blood contains a cancer signal. And after a full workup, scans, biopsies, wait weeks or months, they will learn they don't have cancer. That is not nothing. The psychological cost of being told for a month of your life that you might have cancer, and then learning you don't, is only now starting to be studied as a medical outcome in its own right. There is also the question of what doctors call overdiagnosis. Finding a tumor so tiny, so slow, growing it would never have hurt you. Now you have a label. Maybe a biopsy? Maybe surgery for something that would have stayed quiet for the rest of your life. Early detection is not the same as early cure. A cancer found at stage 1 is easier to treat, but it also has to actually be a cancer that was going to kill you. Distinguishing caught just in time from we operated on something that was never going to matter is the hard part of the next decade of this science. The honest version of this story is, the test works. It finds cancers that would otherwise be missed. It also occasionally tells healthy people they might be sick, and sometimes flags tumors that would have been fine if ignored. Both of those things are true at the same time. Medicine has rarely been able to hold both. Here is the detail about the GRAIL story that I cannot stop thinking about. Laura Huber died on November 10th. Her husband's first meeting about the new cancer detection company was December 10th. GRAIL was announced publicly on January 10th, and Jeff Huber was named CEO on February 10th. 4 dates. All the 10th. He has never explained if that was on purpose. Reporters have asked. He does not answer. Maybe it was grief structuring itself into a schedule. Maybe it was grief structuring itself into a schedule. Maybe it was a quiet ritual. Maybe nobody thought about it until months later, and by then the dates were already what they were. What I know is this. A man in the worst year of his life walked into a room 30 days after his wife's death, and he said, not with bitterness, but with terrible clarity, we are going to build this. 10 years on, there are people reading these words whose cancers have been caught by Galleri at stage 1. People who will live to see their grandchildren because a blood draw flagged a signal their body had not yet noticed. That is what grief, very occasionally, is capable of becoming. Not a monument, not a memorial. A working piece of infrastructure, running quietly in clinical labs across the country, every day, finding a signal that used to be silent, your body right now is writing. Every cell you have is recording a little biography of itself in methylation patterns. Then, breaking up and sending those sentences into your bloodstream. If a cancer is growing somewhere in you, and statistically for some of you watching, one will, someday, those sentences are already being written. In ink we once could not see. For the first time in human history, someone can read them. If what we just talked about felt like science fiction, an invisible signal written for 1 billion years, suddenly readable in a tube of blood, wait until you meet the caterpillar that wears the bodies of its victims. High in a mountain on the Hawaiian island of Oahu, in a hollow spiderweb, a biologist shines a light on something that looks like moving debris. It is a caterpillar, but glued to its back in a careful armor are the severed legs of ants, the wings of dead flies, The heads of beetles. It wears them. It hunts other insects trapped in the web. It kills them, dismembers them, and stitches their pieces onto its own body. Nobody had ever seen it. It had been hiding in plain sight in the state of Hawaii for millions of years. And then, in 2025, a single scientist with a flashlight finally looked closely enough. The universe is stranger and more beautiful than we imagined. It keeps being that way until somebody decides to look.